Welcome everyone to the second webinar that is part of our Phenology Week. We are celebrating spring this week, as well as celebrating all of our Nature's Notebook participants and partners across the country in our celebration of Phenology Week. Today, we are lucky to be joined by several researchers who have used or are using Nature's Notebook data. So I'm really pleased to have them be here today to share with you all some of their research and then also be available to answer your questions about how your data are used and other things that they've been working on in the field of phenology. So before we get started, just wanted to give a little bit of an orientation to our webinar today. So you will be able to post in the chat, and we already have people that are sharing where they're calling in from, so feel free to keep posting that. Also, feel free to put in anything that you're seeing in terms of signs of spring, or maybe not so signs of spring, depending on where you are in the country. We love to hear, reports of what you're seeing around you. We also have a Q&A feature, and so I definitely welcome you if you have questions for our presenters. You can use that Q&A feature. That'll really help us kind of keep track of the questions that are coming in. So definitely feel free to use that. And with that, I think we will start with our presentation. So first up, we have Dr. Lin Meng. She's a global ecologist and assistant professor of Earth and Environmental Sciences at Vanderbilt University in Tennessee. And so I will pass it over to Lynn.
Hello, everyone. Thanks for the opportunities. It's really exciting to be here to be part of the Phenology Week and sharing my work about technology and artificial light with you. So I'm an assistant professor at Vanderbilt University looking at the interaction between plants, climate change, and urban built-up environment. And today I'm going to talk to you why artificial light is important in understanding phenological changes. Plants do not have watches or calendars, but they seem to know when spring arrives better than we do. The timing of this seasonal biological event, such as when trees leaf out, produce flowers, produce fruit, is called phenology. Understanding phenology is really important because plants provide evapotranspiration cooling effect. So if we have a longer growing seasons, we'll have a longer cooling effect provided by plants to mitigate urban heat island and absorb more carbon dioxide from the atmosphere. Also, the phenology is highly correlated with pollen seasons. So early phenology could mean early and more severe pollen season for pollen allergy group. Also, phenology will also affect energy use and our utility bills. So understanding the changes in phenology is really the key to understand all these other things that we care about. And phenology involves a really complicated process, but during several decades of research, scientists figure out that temperature and day length are two of the most important environmental cues for trees to track time. So in winter, when temperature decreases, tree will go dormant. And then this is their way to prevent damage from harsh winter. And then as trees experience a period of cold temperature, we call it chilling period. They break dormancy and start a countdown to spring. And then as days get warmer, we see trees with green leaf emerge. But temperature is highly fluctuated over years. So only depending on temperature would be kind of risky for plants. So they also depend on the natural day length because the length of the day is a very reliable signals for seasonal progression. It doesn't change over years. So temperature and day length, say, both of them are environmental triggers for trees. However, in cities, both triggers have been largely altered because of the urban built-up environment. We have urban heat island, cities often 1 to 3 degrees warmer than the surrounding countryside because we have buildings, parking lot, all these materials that can absorb and retain heat. So urbans are warmer than rural areas. We also have artificial light in cities. Nowadays, millions of children that cannot see Milky Way where they live. Those light that come from the office block, homes, street light, and vehicle light, they do not just light up what we need. They spill into the habitat of plants and animals. And those light largely alter this natural day-night cycle that plants adapt to. And there's already enough evidence from the plant physiology to show that light have a profound impact on plant development and phenology, because there are photoreceptors that have been identified in plants that can sense different intensity of light and color of light. For example, this low pressure sodium light, this yellow light, sensed by one type of photoreceptor, and this blue-rich LED light, which is a more energy-saving lighting strategy, are sensed by another type of receptors. So by changing the intensity of the light and the color of the light, it will affect plant phenology, but this is largely unexplored. So my research is to fill this gap to understand how urban heat island and artificial light affect plant phenology along this urban-rural gradient. So I look at phenology for the same species in urban versus in rural, and then I examine the temperature level and the artificial light level to figure out how the extent of light impact on plant phenology. Previously, we don't have many research focusing on artificial light impact on phenology, because we don't have enough phenology observation in urban areas. So in my research, I use USA National Phenology Network and Nature's Notebook of phenology data in cities, so that I was able to conduct this research. The artificial light products I use is a most recent one from NASA called Black Marble. This is a daily artificial light product, and I only focus on the site that I have phenology observations. So you can see the brightest regions are cities and some transportation network. And then I selected 2 phenological events, breaking leaf bud and color leaf from Nature's Notebook. And then I overlaid my phenology observation and the artificial light map. So you can see those green dots are the locations I have breaking leaf bud observation. And this orange dot are the one that I have color leaf observations. So what I did is I classified all my phenology observation into 2 categories. Phenology site with artificial light shown in this red line, and phenology site without artificial light shown in this blue line. And I examined the phenology date for these 2 categories along every 1 degree in temperature. So in this way, I can disentangle the effect of temperature and artificial light. And then I found, on average, those sites with artificial light show 9 days earlier in breaking leaf bud. This is pretty profound. This is more than a week. For colored leaves, it's more complicated. When temperature is below 23 degrees, I found artificial light delayed autumn colored leaves. But when temperature is higher than 23 degrees, there's more fluctuations in the data of phenology. And based on this finding, I built a linear mixed model, and then I used future temperature projection and 3 artificial light scenarios in the future to predict the changes in breaking leaf bud and color leaves in the future by the year 2100. And I found the under climate warming, phenology show a continuous earlier trend, but on top of that, artificial light will shift phenology much earlier by 7 days, which account for 40% of climate warming effect. For color leaves, there's a non-linear effect of artificial light on color leaves. It will delay the color leaves first and then advance it. So the take-home message is we found a significant artificial light impact on plant phenology. So what this means is when we talk about sustainable cities, nature-based solution for urban green infrastructure, we want to have a full pictures. So the artificial light and especially minimizing the ecological impact of artificial light need to be part of the solution. I would like to acknowledge all my co-authors in this paper, and if you want to learn more about details, feel free to check out papers. And our work inspired a poem called Artificial Rhythm, posted on the Poetry of Science, and we were interviewed by several medias, including NPR. So it's really exciting to see that the media and the public are very interested in understanding the ecological impact of artificial light. That's it. Thank you for your listening, and I'm happy to take any questions.
Thanks so much. It's so interesting to learn more about your research. It's really an understudied area, and so neat to see that you're able to use both the breaking leaf bud and colored leaf reports that our Nature's Notebook observers have been reporting. So thank you so much for sharing about that. I don't think we have any questions yet, but feel free, if anyone has any questions for our presenters, you can put those in the Q&A, and then we'll have time at the end for questions. So if no one has any right now, we'll keep moving along through our presenters, and then we'll get back to questions in a little bit. So thank you so much, Lynn. So our next presenter is Abbie Anderson. She worked for the last 3 years as the program coordinator for Pesky Plant Trackers, which was a campaign that we did with in collaboration with the University of Minnesota. And she was also the coordinator for Cover It Up, another citizen science program, both based at the University of Minnesota's Department of Forest Resources. And Abbie is also a team member for a program that's called Season Watch, and they have a website that's all about phenology in Minnesota. So I'll put links to those in the chat while Abbie presents. So I'll pass it over to you.
Thanks so much, Erin. Thanks everybody for making time to join the webinar tonight. Today, I'm really excited to have this opportunity to talk to you about pesky plants and how we use Nature's Notebook to inform management of invasive plants. Funding for pesky plants came from the Minnesota Invasive Terrestrial Plants and Pests Center through the Minnesota Environment and Natural Resources Trust Fund. And the staff for the small team were all with the Department of Forest Resources. And we also worked with a number of other organizations and state agencies in the state of Minnesota, as well as our volunteer observers, of course, contributed a great deal to make this possible. I'm going to introduce our research team quickly, and I want to emphasize that we come from a lot of directions to synthesize all these different kinds of interests and areas of knowledge to make our team work. So our principal investigator is Rebecca Montgomery, a forest ecologist. Baiju Govindan is a data modeler on our team. Stephen Carlson is another researcher on our team. And then there's me, and I'm the program coordinator, and my specialty is that circle in the left, participatory science. I'll introduce the plants that we studied. We studied, as I said in the title, it's all about invasive plants. So the two plants we studied are regulated noxious weeds in the state of Minnesota. They come from other parts of the world and they do cause harm in Minnesota. The first plant is wild parsnip. This is a map of where it's reported in North America. And as you can see, it's been reported in most of Minnesota. And like the other invasive plants, I'm sure many of our listeners know about, they displace native biodiversity, both plants and animals, and degrade the quality of habitat for the biodiversity that we'd like to see here. Wild parsnip is also a dangerous plant for human health. It causes skin burns that can be quite severe. So if you see this plant, don't touch it. The other plant we studied is Japanese knotweed, as well as some of the related hybrids. And this plant is reported both in the Northeast and in the Northwest, and within the state of Minnesota is moving from east to west. This is also a plant that causes harm in the environment. What you see in the top right is an aerial photo of a property where knotweed has invaded, and it's that orange color. In the bottom of this slide, that's a picture of a stem of knotweed that has emerged inside of a basement. So this plant has the ability to break through hard surfaces and cause costly damages to infrastructure and buildings. So these are our plants that we're studying because ultimately our goal is to remove them from the environment where they're causing harm. So we want to learn more about them in order to inform management. There's a lot of different actions that land managers have in their toolbox, but they're all time sensitive. And that if you consider the life cycle, this is a life cycle of wild parsnip on the right. You have all these toolbox of different actions you can take, but in order for them to be effective, they need to coincide with the phenology of the plant. And this is just as it's true for wild parsnip, also true for knotweed in this diagram. And so we use this kind of as the taking off point for what we wanted to study. Because we knew that weather drives plant phenology, which then informs the timing of management actions. But if you look at this framework, the piece that's missing in terms of data is the phenology data. So our goal was to get those phenology data, then model the data and share those models with land managers to improve the timing and effectiveness of invasive plant control. We wanted to share the models at this website called uspest.org that serves phenology and degree day models. So it essentially lets land managers put in a zip code and calculate when a plant of interest will reach a given phase. That is when it's most vulnerable for taking management action. So we wanted to solve this problem of helping land managers schedule their work. The methods we use is that we use the protocols and tools developed by USA National Phenology Network. We use Nature's Notebook, and as Erin mentioned, we created a campaign called Pesky Plant Trackers. And it was the phenophases that we wanted to collect. These are just some of them for wild parsnip: initial growth, flowers and flower buds, open flowers. Not pictured are fruits and ripe fruits. And similarly, we wanted to collect that kind of information on Japanese knotweed, pictured here. So how are we going to get that phenology data? We knew that we wanted to get a lot of data in the field, so the plants were actually experiencing the naturally occurring environmental conditions across the state of Minnesota, and we wanted a big geographic range. That was really important to us. So one of the ways we did that is we worked with the university's system of research sites to set up some experimental plots, and we had 15 really high intensity volunteers who monitored plants at these sites. I say high intensity because they monitored not just the phenology of 1, 2, or 3 plants, but about 30. And they did that every week for a couple of years. Additionally, we had about 150 volunteers who did what I called opportunistic sampling. So that's where these plants are already occurring in the environment. That's an opportunity to monitor and collect data. And we had a terrific community of volunteers who took that upon themselves and collected a lot of data across not only Minnesota, but also beyond. We also, in addition to the data in the field, we had efforts to collect data within growth chambers where we could manipulate the temperatures. We did some seed germination trials, and we did what's called a cold chamber or vernalization trial for wild parsnip, because that chilling period is important in its overall phenology. But I'm mostly going to talk about the data in the field. And having talked about all that data collection, I'm going to actually skip over data modeling for the moment and just reflect quickly on participatory science, since that's my area. And so I want to just talk about tea. Well, I'm sure tea is important. I'm sure phenology is important for tea, but I want to talk actually about a program we did called Tea Tuesday. When we started Pesky Plant Trackers, it was 2020, so it shouldn't surprise you that we were working with volunteers, reaching out to them by Zoom. And Rebecca, the principal investigator, had this great idea that was really instrumental in the success of the program, that I hosted a Zoom every week during our field season and invited our observers to come in and spill the tea. Tell me what they're seeing, what are their plants doing, are they seeing anything unusual or different from what they had expected, and we would share guidance on how to interpret what we're seeing in the field. And so through this small network that joined for tea, I was able to then, by the exchanging of information horizontally across that small group, I could then share that out with our larger group through the website and the newsletter and help people interpret phenophases that they were seeing out on the land. I want to thank everyone who not only collected data out there, but especially people who joined tea and made the program really possible through sharing all that information. I'll speak just real quickly about modeling the data because this is in progress. But one of the things that our data from the field lets us do is understand phenology in terms of cumulative degree days. So that cumulative degree days is a way of saying how a place is warming up over time and how the plants are responding in concert with that. Specifically, one of the questions that we are modeling is how many cumulative degree days need to pass before 50% in a given 50% of the plants in a given location reach a given phenophase. So I'll share a couple plots that look like this. On the x-axis, that's the cumulative degree days. So you want to track how many degree days are accumulating, and then in response, what proportion of the plants are reaching a phenophase, so that you can calculate how warm it needs to get before you see the change that you're interested in. So at this point, because the models are in development, we have a number of plots and each one represents a different year in a different place. So we're synthesizing all this information. So I'm going to give you a kind of peek into that process. So for wild parsnip, for example, initial growth, we're calculating the cumulative degree days needed to reach that phase. Also for flowers and flower buds and for open flowers. Similarly, we're doing these calculations for knotweed, initial growth, flowers and flower buds, and open flowers. And one question I could anticipate people asking, well, which of these different numbers highlighted in yellow is the one I should pay attention to? And the answer is because of the way we collected the data, it actually adds dimension and robustness to the data modeling so that we can actually answer that in a variety of settings and a variety of environmental conditions. So when the models are finalized, and these are just kind of progress states, they will be online at uspest.org. And this is a freely available tool for land managers to use to schedule their work. And so we come back to these diagrams, how we're bringing more detail in a very site specific and case specific so that people can use this diagram to predict and make forecasts. We also are engaging in some conversations about more general, what did we learn about the biology and ecology of these plants? Because we certainly had a lot of interesting things observed. So those conversations are also going on right now in our expanded team, who we're working with to kind of just discuss what we found and assimilate it into the larger body of knowledge. That's what I have for pesky plants today. Thanks so much for coming to the webinar.
Thank you, Abbie. It's such an amazing project that just covers so many different dimensions of what we do, from the practical applications of how you can actually use phenology to improve invasive species management. And then I just love your insights on working with volunteers and providing that network through Tea Tuesday. And we've also taken one of your other ideas, which was having a gratitude party. So just putting in a plug for our Friday webinar, we're going to be hosting a gratitude party for all of our Nature's Notebook observers at the same time on Friday. So make sure you register for that as well. So I think if we had any specific questions yet for Abbie, I know we have some questions for Lynn, so I think we'll skip and have those at the end after the next presentation. But I think, let's see. Well, yeah, we'll go to our final presenter and then we'll get back to questions. So our final presenter is Dr. Kerissa Battle. She's a research ecologist and a social entrepreneur who founded the Community Greenways Collaborative and also is the founder of the New York Phenology Project. And Kerissa has been working with us since, we just decided, since 2010, so a really long time. So I'm happy to pass it over to Kerissa.
Hi, everyone. Thanks for having me. So I do want to start out with some extra gratitude. Number one, oh, actually my slide isn't advancing. Okay, that worked. Okay, so to the observers, which of course we would not have any data to be working with if we didn't have all of you incredible observers out there. And then some extra gratitude to NPN, who, as Erin said, we've been on this journey together for a while, and it's been this really amazing thing to witness the mutuality between the observers and participants and NPN, and how the feedback loop has worked to really mature the community over the last 15 years. It's been a really incredible thing to watch, the responsiveness from NPN, and just the extra time and effort that the observers take to not only do their observing work, but to help the team at NPN grow all of the resources that we use to make this work happen. So happy 15 years, NPN. So before I get to the research, just another thing that I've been able to witness as both a researcher, participant, observer, volunteer observer, and also somebody who's been involved in helping launch some of the networks and projects, particularly in New York, first in Oregon and then in New York, is that I've just seen the participation really, it's more than just the research. It really strengthens the bioregion. Sites get connected to one another, whether you're a backyard observer or working at an institution. I've seen a lot of training nodes happen where the various nodes in the network can self-replicate, so it doesn't require a ton of resources. Lots of new kind of projects, land management, that both the data and the activity of the community doing the data collection can lead to all these this diffusion of innovation around phenology monitoring. Of course, the educational curriculum. I think the impact also on people individually, not to discount that mindfulness practice, and just the personal health, getting outside, connecting with nature, and just being slow and having that opportunity to interact with organisms as you watch them through their life cycle is actually, I think, an incredibly enriching thing to be doing, even on a personal level. So, so watching all of these incredible things take place over the years, it's been really wonderful to witness. And of course, the research potential has been incredible, and watching the research mature over the years has also been an amazing thing. More and more publications out there using citizen and community science data. So I'll start here with the research. So in 2012, I launched the New York Phenology Project and started creating these sites, or helping launch these sites across the state. And it was going well. We were collecting data, but of course, you know, it's going to take a while before we can do any analyses. And one day out of the blue, I get a phone call from a local naturalist who tells me that he's uncovered a data set from the early 1800s, which looks very similar to the data set being collected by Nature's Notebook observers in the 2000s. And as you can imagine, as a researcher, this was like an existential experience to have somebody tell me that there was a comparable data set that existed from 200 years ago, monitoring the same plants in a very similar way across the entire state of New York. Like, how did that happen? I don't know, but obviously I was pretty excited about that. And we started to collaborate this is the Hawthorne Valley Farmscape Ecology Program. The researcher who called me, the naturalist who called me, was from there. So we digitized this raw data set, standardized the protocols between the two time periods. As you can imagine, the language wasn't totally consistent, you know, given these records from long ago. But the protocols, the description of the protocols matched really well. And so we pulled these two data sets and then started to make some decisions around what to include, what not to include. We had a lot of decisions to make, you know, with what species. We went from over 900 species down to 36 that we felt we could really match with the kind of robust numbers that we wanted in terms of how many years of data and number of observations. We ended up between the two pooled data sets with 150 different locations across the state, 12 forbs, 12 shrubs, 13 trees, and then took the data from the past and from the present and did a comparison. And we used the two phenophases we used were breaking leaf bud and open flower. And we used these folks back in the 1800s also collected weather data. So it's paired, one of the earliest known weather data collection efforts in North America. And we had paired phenology and temperature data to compare both in the past and the present. And so we used January to April temperatures, and we asked these 4 questions. Basically, how has the flowering leaf phenology of these plant species changed over the last 200 years? Are the changes associated with warming temperatures? Which species or groups of species are responding most and least to changes in climate? And then is variation among phenology related to seasonality? So whether, you know, early spring, late spring, life form, trees, forbs and shrubs, urban rural areas, and pollination syndrome, which was only for trees, wind pollinated and insect pollinated trees. So we found that the majority of the plants are, as you would probably guess, leafing out and flowering earlier. Overall, on average, flowering by about 10.5 days and leafing out by 19 days. We did find that the temperature since the first record since 1820 has advanced by 2.5 degrees Celsius. And that the plants are leafing out, both leafing out and flowering, about 3 days per degree Celsius change, both from the historic to the contemporary time. Here's just some shout outs for the plants here that we noticed flowered or leafed out advanced the most. As you can see, you know, some advancements up to 27 days, which is, you know, quite a difference between the past and the present. As you can imagine, some key plants like milkweed or red maple, both really important pollinator plants, both about 2 weeks. And we did notice some differences between, the trees and the forbs, which could theoretically complicate, the canopy light differences and how forbs respond. So lots of possible issues with this level of difference in timing across life forms. And we did find that the variation among phenology related to seasonality, that early season species are tending to advance more. We did find more differences for trees and shrubs more than forbs, that urban areas, as you would imagine, are advancing more rapidly, and that insect pollinated trees are also advancing more than wind pollinated trees. And so because of the amount of data that we had and the quality of the data we had, we were really able to answer these broad scale questions. And I think that this study is an example of, obviously really important and good to have this kind of science information, but I think the study itself was also an example of what is possible when you have networked monitoring across an entire region. And it really, there's been so many great impacts from this study, from new curriculum and new sites, new observers, and we're grateful that we had the opportunity to do this study, and thank you again to all the observers that collected the data. I'll stop there.
Awesome. Thanks so much. What a gold mine to find that data set. It just must have been so amazing. And also because of the great data set that you already had collected through the New York Phenology Project, you're able to do that comparison. So it's a testament to all of your hard work in getting all of those local phenology programs set up and making sure that they have all the resources they need. So we really appreciate regional coordinators like you that are coordinating so many different people across the whole state.
Thanks, Erin.
So we do have some questions. I think we'll go back to some questions for Lynn. So we had some questions that were in the chat and then one in the Q&A as well. And there were a couple questions about your advice for people who are trying to grow or support trees or plants that are growing in urban locations. Is there anything that they can do to kind of help the trees? Is there a particular kind of light that's best, or practices where they can try to avoid harmful effects of artificial light?
Thanks for the question. So the impact of artificial light on trees is really complicated. There are some positive aspects and there are some negative aspects. For example, because light is energy, it's food for plants to do photosynthesis. So if there's additional light at night, those light can be used for plants to do photosynthesis. So there are already some studies that observed those trees that's next to or closer to artificial light will have bigger leaves because there's more food. So trees will use this light to do photosynthesis and grow biomass. But it also disturbs the tree phenology. And they may have some other impact. For example, it may change the evapotranspirations, but we don't know yet. So right now we know very little about how artificial light impact trees in cities. This is definitely need more investigation in the future. But different tree species have different sensitivity to artificial light. So those trees that are less sensitive to light, that are more sensitive to temperature, maybe would be a good candidate for street trees or city trees that will mitigate artificial light impact on trees. And also there's another questions about what are the best practice for outdoor lighting. So there is Dark Sky Association that they provide some guidance of what can we do to minimize the ecological impact of artificial light. But basically, warm light is good for animal and plants compared to white light, because white light has, especially this LED light, has a broader spectrum, so the more affected, they will affect animal and plants more. And also, it's recommended that we only turn on the light when we need, and we only use the necessary brightness of the light.
Great, those are some good, simple actions that people can take. So thank you. And then we also had a question for Abbie about what is the best way to try to limit these exotic plants, in particular wild parsnip and Japanese knotweed, but I know that's part of the research that's ongoing, is trying to figure out the best timing. But what do you recommend if people come across these plants now? What's the best way to get rid of them?
Well, there are some best practices that I would point to, like your state Department of Natural Resources will have information about mostly not transporting plants. That is the most important thing. So when you're walking in a natural place, use a boot brush before and after you go places, and just be aware of seeds that can attach to shoes and clothing. So I think number one is to be familiar with those best practices about not spreading plants. If you find them growing, then there are systems that you can use to report plants that might be of interest to a county agricultural inspector. And I've actually, so there's a system called EDMAPS. It stands for Early Detection Distribution Mapping, and it's really pays attention to all kinds of invasive species. So if you're really interested in being proactive and building the information to help people be aware and improve environments, I would say share that information with EDMAPS. We can put a link to EDMAPS in the chat. Thank you. You did that already.
Yeah, thank you. All right, so feel free to keep putting your questions into the Q&A box. I did have a couple questions that maybe we can do kind of a round robin, but I'd love to hear from all three of you about how did you first become interested in phenology, or if you remember when you first heard the term phenology, that would be great to know as well. I remember that it was when I started working at the NPN that I first heard the term, and even with a background in environmental biology, I'd never heard the word phenology before. So anything you want to share on that? Maybe we'll start with Kerissa.
Sure. So in my ecology training, I mean, I think it started out, doing these big surveys where you're learning all the names of the plants and going vast distances and covering lots of ground and all of that. But it wasn't until I was living in Southern Oregon and walking the same trail every day that I started to really form these deeper relationships with the plants because I was watching them through their whole life cycle. And it sort of changed my mindset in a way from kind of that, wanting to know everything about everything to getting, more focused on this longer term relationship with plants. And then that led to thinking about this idea of a phenology trail and then getting in touch with NPN and the rest is history. Mm-hmm.
Awesome. Maybe Lynn, do you want to share next?
Sure. So I got interested in phenology because of personal experience looking at the cherry blossom. When I was doing my master in 2014, like 9 years ago, and there's a spring that my friend and I plan to go to the cherry blossom festival. But the night before we went, a snowstorm came. So what we saw on the next day was a complete loss of all those emerging blossoms and just dead flowers all over the ground. So that experience made me really think about what will affect the timing of flowering and the leafing out. Because I realized in the spring, plants are really vulnerable to these changes in the environment. And if they start to leaf out and flower too early, then they are at this late spring they're facing this risk. But if they are flowering too late, they'll miss opportunities to do photosynthesis and grow themselves. So I thought this is really interesting research questions. So I was looking for a PhD program that can allow me to study this, and I went to Iowa State and then decided to look at phenology and urban environment as my PhD dissertation research. Yeah, that's how I got interested in phenology.
Awesome, thank you. What about you, Abbie?
I love this question because I actually came to, I don't remember how I first heard the word, but I came to phenology really as an artist. I was working in an art museum that had a sculpture garden, and I was enjoying the possibility of interpreting that space by looking at ephemeral events that happened in nature, especially as a nice backdrop or kind of contrast to sculptures, which are pretty static and unchanging over the year. And then in contrast to the fact that the environment is like a different environment every day. So I started a project called Open Phenology that was very open-ended, small conversations all around interpreting nature and ephemeral events in the sculpture garden. So that's how I first started. And that was over 10 years ago.
That's amazing. We have another question in the Q&A that I think probably is best suited for Kerissa, and that is, have any examples been found where flowering of a given plant is becoming out of sync with the life cycle of its specialist pollinator?
Yeah, there's definitely some research out there. Gary Kudo has some research. There was just a newer one published in 2023 from England, I believe, but that was a really good, sorry, all on my computer. And that one's called Phenological Mismatch in the Demography of Solitary Bees. And that was a good paper that demonstrated specific mismatch. So that would be a good one to go to. But yeah, I mean, there's definitely examples out there of mismatch, although it's not consistent. You know, not every study finds the same trends and patterns. And as with everything in ecology, I think, you know, depends on where you're studying and the exact organisms you're studying and how many seasons of data you're collecting and all of that. So, but yeah, there's definitely examples out there. Queen bees, bumblebee species are being found to emerge earlier and being out of sync with nectar resources. And yeah, there's a fair amount of examples out there. Mm-hmm.
Thank you.
Yeah.
And then a question for Abbie, are there plans to expand your program to other invasives such as common tansy?
Great question. We are open to that. It's our project was funded by the Minnesota Invasive Terrestrial Plants and Pests Project Center. So it would be, you know, we are in conversation about like what's the next target species that phenology could have an important place in solving real world problems. So that's under conversation. It's not in the planning right now, but you're exactly right. It's this kind of principle could apply to other things. So, and Rebecca added to the chat there about that. So,
oh, great. Thank you. So another thing that I would like to ask you all is just thinking back about your experience either using the phenology data from Nature's Notebook or working with volunteers, part of Nature's Notebook. What was the most challenging part of working with the data or working with volunteers, or maybe the most inspiring if you don't have any challenges? But anything you want to share about that? Maybe we'll start with Lynn first.
Yeah. I would just say the general challenges of doing phenology research, because my research is specifically focused on urban environment, and I didn't start it with USA National Phenology Network and the Nature's Notebook. I started with using remote sensing phenology product, and our current satellite product doesn't have enough coverage in cities, and there's many missing values in cities. So it's really, there's a large uncertainties in using a remote sensing product. So, and then the other thing is satellite product cannot differentiate different tree species. So there's always a mixed information of multiple tree species in one grid. And tree species is really important because species can have very different phenology even under the same temperature or light conditions. So that's why I kind of looking for other alternative data set, and I found there's a USA National Phenology Network and Nature's Notebook. And because it's collected by citizen scientists, so it's mostly in cities and in some nearby regions, and it's species specific. So this data set perfectly solve all my issues. So I started to use this data set and to develop this light pollution project. So I would like to, acknowledge data observer and collectors thanks for your hard work, because this data is really valuable for researchers like me to look at how phenology responds to climate change and urban environment. And then I spent some time trying to understand the data protocols and the specific phenological events that I'm going to use for my research. But I think there's a very detailed description and guidance online. So I was able to just follow that and to figure out the specific phenological event and then select that as a subset of data and use that for my research.
Awesome. Abbie, do you want to share anything about challenges or inspiring things about working with data or volunteers?
Yeah, I have one of each. I'd love to. Challenges, I would say what comes to mind is actually a bit of a communication challenge, because we worked with invasive plants that are generally not desired, not preferred in the environment. And so we had a challenge of like, trust me, let's just watch them, but not touch them, not remove them. But how do you also say it's important to remove them while also we're going to just watch them without interfering? So that was a little bit of a communication challenge. And then what was most inspiring is easy, like the volunteers, and it's so interesting. You come across someone who they're like, yeah, I want to do this project. And I have to say like, well, tell me more about why you want to do it, because it's so interesting to me. Everybody's stories is different, and I can learn a lot from what people's encounter with these plants are, to then amplify that experience with a bigger community. So that was really fun.
Thanks for sharing that. What about you, Kerissa?
Well, I think the challenge really was just sometimes the heartbreak of working in a resource limited environment. I mean, I think so many of us in the environmental field experience that, but there's no shortage of really passionate people and volunteers available to do the work. And then, organizations sometimes get started and then they can't continue because they can't fund a volunteer coordinator, or so there's just over the years have seen communities kind of form and then not be able to continue because of resource limitation, or because it's difficult to prioritize long-term monitoring from a kind of policy level, especially if there aren't these really obvious direct ties to management. So I am so appreciative of folks whose work is really being tied to management, because I think that will keep the resources coming in for the long-term monitoring scene. So obviously, on the flip side of that, seeing all these communities and being part of them has been incredibly inspiring and enriching. As I mentioned before, just watching that a new level of nature connection take place, and that the benefits of that for people's lives forever, not just while they're participating in Nature's Notebook, but almost as it's going to be like a family legacy for future generations to have that kind of new new but old relationship with plants and animals. You can tell I'm a plant person. And yeah, just people's dedication and why they're out there spending time doing this. It's an incredible thing to see. So thank you so much.
Definitely. So yeah, and we also have some observer stories that we've been sharing this week that are really inspiring, just hearing about what doing observations regularly means to people. So definitely check those out on our social media. I'll put a link to our Phenology Week landing page in the chat. You can sign up for daily emails. Oh, and Samantha for just put in the chat too. If you can also sign up for emails where you get an email each day this week, and we're sharing those stories there as well. So I know we just have a couple minutes left, but just wanted to offer a chance to, do any of you have any advice for our Nature's Notebook observers that are on the call today? Anything that you'd like to say, kind of words of wisdom or things from the side of using the data that you could offer back to observers that are collecting the data? And I'll just leave it open if anyone wants to jump in.
I'll offer something. First, don't touch wild parsnip. We had a really good safety record in our volunteer community, so thank you. And I say, trust yourself. Trust what you see, because sometimes you see unexpected things.
It's great advice.
I would just say enjoy every minute of being out there and building your relationship with what you're watching and sharing it with everyone you know. Don't be afraid to be super nerdy. Everyone likes it more than you think.
I would say like long-term observation at one specific location is really valuable for research. And if we have more observation, like for the same locations, that will be great.
Yeah, that's wonderful. And also putting in another plug for our webinar on Friday, we're going to be highlighting some of our observers who are the most long-term observers that we have participating. And some of them have been collecting data for 15 years or 14 years. So we appreciate anyone who's participating as part of Nature's Notebook. But as Lynn said, those long-term observations collected at the same individual plants or the same sites over time are just so valuable. So if you are a Nature's Notebook observer, we hope you stick with it for the long term because your data are just so valuable. So thank you all so much for providing all of your insight, telling us about your research and answering questions. It's so valuable, I think, for our observers here today to be able to interact with you all and kind of get that personal perspective on talking with people who are using their data. So really, thank you so much. I think I'm going to pass it to Samantha Brewer before we leave, and she's going to share some other events that we have going on this week for Phenology Week.
Hi, everyone. Thank you so much for being here with us today. And it's just wonderful having you. And thank you so much for our researchers who are able to join us and talk about all the work that you've done and your inspiring message. That was just phenomenal. Thank you. So yes, this week we have a few more events happening. Tomorrow we have our Plantside Chat video with Kerissa Crimmins that we're going to be releasing. So if you'd like to hear from our director a little bit about what she has to say and about what some of the work that's happened from your efforts, feel free to join us for that. On Thursday, we'll be announcing our 2022 Pheno Champions, and we'll be discussing our annual report and our local phenology program annual survey results. We send out a survey every year from local phenology programs to hear about what they've done and how they've used our program and what they've learned from your efforts. And then on Friday, March 24th, we have our gratitude party virtual celebration to celebrate everything that we've learned from all of you. And we'll also share anything that you've shared with us this week about your phenology observations, or if you've answered any of our week or our daily challenges for Phenology Week. We'll be sharing those on Friday as well. So we welcome you to join us for any of these events.
Awesome. Thank you so much. And thanks everyone for joining us. We will see you online the rest of this week. Thanks a lot.
Thanks, everyone.
Thank you.
Bye-bye.
Thank you. Bye.